We're following efforts to clean up the Chesapeake Bay so future generations can enjoy it for many more years to come.
They called it a dead zone for a reason. Nothing could breathe there. If you swam in it, you were swimming in a graveyard. But in 2012, a group of researchers did the unthinkable. They released 10,000 juvenile blue crabs right into the heart of the pollution. It felt like sending soldiers into a battle they couldn't win. But here's the catch. We completely underestimated what these creatures are capable of.
Scientists explain that the best way to understand any organism is to understand its genetic blueprint. So they say that they've learned how it functions, including which genetic traits make crabs particularly successful at reproducing or adapting to their environment.
What started as a simple release turned into a biological revolution that shocked the entire scientific community. The toxic graveyard beneath the surface. The Chesapeake Bay is massive. We are talking about a body of water that stretches 200 miles, covering nearly 4,500 square miles of surface area. To put that into perspective, that is bigger than the entire state of Delaware. For centuries, this wasn't just water, it was a gold mine. People called it the Great Protein Factory. Watermen would go out and pull 20 million bushels of oysters out of the bay every single year. That is enough oysters to fill the United States Capitol building twice over. It was an endless supply of food, money, and culture. But fast forward to 2012, and that paradise had turned into a nightmare. Something had gone catastrophically wrong. The bay that once fed millions had transformed into what scientists politely called an aquatic dead zone. But let's be real about what that actually means. It means the water was so toxic that fish were literally swimming away to die somewhere else because they couldn't breathe. The bottom of the bay had become a graveyard. The blue crab population, the signature species of the region, had collapsed by over 70%. So here is the deal. The Chesapeake Bay is fed by over 150 rivers and streams. It drains parts of 6 different states: New York, Pennsylvania, Delaware, Maryland, Virginia, and West Virginia all dump their water here. Everything that happens in those states eventually flows into the bay. Every farm, every factory, every sewage treatment plant, and every suburban lawn drenched in fertilizer ends up right here. It is like a giant funnel for the entire mid-Atlantic region's waste. Starting in the 1950s, industrial farming exploded. We aren't talking about small family gardens. We are talking about massive operations pumping millions of pounds of nitrogen and phosphorus into the soil. Plants only absorb maybe half of that fertilizer. The rest, it runs off into the streams and heads straight for the bay. When nitrogen and phosphorus hit the water, they don't just disappear, they act like super fuel for algae. This is where it gets scary. Algae blooms sound harmless. Plants are good, right? Wrong. When these massive blooms die, they sink to the bottom and rot. That decomposition process sucks oxygen out of the water like a vacuum. It creates conditions where there is less than 2 milligrams of oxygen per liter of water. Fish need at least 3 to be comfortable. Crabs need even more. Imagine being locked in a room where the air is slowly being pumped out. That is what every creature in those zones was experiencing. By the early 2000s, these dead zones were appearing every summer like clockwork. Some stretched for miles, nothing could live there. But the fertilizer wasn't acting alone. You had sewage plants dumping waste, urban runoff carrying motor oil, and sediment clouding the water so badly that sunlight couldn't reach the bottom. It was a perfect storm of destruction, and the one creature that could have helped, the oyster, was basically gone. Remember those 20 million bushels? The harvest had dropped to less than 200,000. We had lost 99% of the bay's natural filters. Without oysters to clean the water, the blue crabs were next in line for extinction. These aren't just any crabs. They are aggressive, beautiful scavengers with bright blue claws. They are the apex predator of the bay's floor. They eat everything. But the grass beds where baby crabs hide were gone, choked out by sediment. The water they needed to breathe was empty of oxygen. The females weren't producing enough eggs, and the ones that did hatch weren't surviving. The entire life cycle was breaking down. It looked like the end of the road. The ecosystem was stuck in a downward spiral that nature couldn't fix on its own. The water was too dirty for the animals to survive. And because the animals were gone, nobody was there to clean the water. It was a catch-22 of the worst kind, and that is putting it lightly. Scientists knew they had to do something drastic, or the Chesapeake Bay would become a permanent wasteland. But nobody knew where to start, until one researcher had a crazy idea. The solution seemed impossible, but they had no other choice. Designing the ultimate underwater soldier. Dr. Anson Hines from the Smithsonian Environmental Research Center wasn't interested in small fixes. He proposed something that sounded completely backwards. Usually, when you want to restore an ecosystem, you clean the water first and then you bring the animals back. It makes sense. You don't buy a goldfish and then fill the bowl with bleach. But Hines asked a different question. What if we reversed the order? What if we flooded the bay with juvenile crabs and let them help fix the ecosystem? The logic was actually brilliant, even if it sounded risky. Blue crabs are what ecologists call a keystone species. They aren't the biggest creatures in the habitat, but they hold the whole system together. They are the janitors, the security guards, and the maintenance crew of the bay all rolled into one. They eat dying fish and decomposing matter, recycling nutrients before they can fuel more algae blooms. They prey on species that would otherwise overpopulate. They churn up the sediment on the bottom, which helps aerate the soil. But the criticism was loud and immediate. People thought the scientists had lost their minds. You are going to take 10,000 baby crabs, creatures that already have low survival rates, and throw them into toxic sludge? That isn't restoration, that is a doomed mission. Critics argued it was a waste of resources and cruel to the animals. Why throw good money after bad? If the water eliminated the wild crabs, it would definitely eliminate the raised ones. But here is the catch. The crabs they were planning to release weren't your average bottom feeders. These were lab-grown super soldiers. They were late-stage juveniles, almost adults, meaning they were past the most vulnerable part of their life cycle. They had been raised in controlled hatcheries, fed the perfect diet, and protected from predators their entire lives. These crabs were strong, healthy, and ready for a fight. The team chose their battlefield carefully. They picked the Lafayette River, a small tributary on the Virginia side of the bay. It was polluted, yes, but not total acid. It had decent salinity levels and a few remaining patches of underwater grass. It was the perfect testing ground. If the crabs could survive anywhere, it would be here. But it was still a massive gamble. Nobody had ever attempted a release on this scale in such poor conditions. They had to time it perfectly. Release them too early, and the predators would eat them all. Release them too late, and the water temperature might drop too low. It was a logistical nightmare. They had to transport 10,000 living creatures, keep them stress-free, and get them into the water simultaneously. Each crab had to be measured and tagged. The tagging process was crucial. They needed to know if the crabs they found later were the ones they released or wild ones. They used tiny markers that would stay with the crab even as it grew. This wasn't just about dumping animals in the water. It was about gathering data. They needed to prove that this crazy idea actually worked. There was a lot riding on this. If it failed, it would be a public embarrassment and a blow to conservation efforts. Funding could dry up. The bay could be left to rot. But Dr. Hines and his team believed in the resilience of the species. They believed that nature, if given a fighting chance, could push back. They were banking on the blue crab's aggressive survival instinct. These animals are fighters. If you have ever tried to hold one, you know they don't give up easily. So the plan was set. 10,000 crabs, one polluted river, a team of nervous scientists. They were about to challenge the accepted wisdom of marine biology. They were about to see if an army of scavengers could do what millions of dollars in technology failed to do. The trucks were loaded, the boats were prepped. Everything was ready for the moment of truth. The day the river woke up. On a humid morning in June 2012, the boats motored out into the murky waters of the Lafayette River. The air was thick and heavy, much like the water below. Researchers worked quickly, lifting crates of scrambling, clicking crabs. 10,000 of them. One by one and in bunches, they were tossed overboard. They sank into the dark green gloom and disappeared. Then the waiting began. And let me tell you, the silence was deafening. The first week was pure tension. Had the crabs immediately fled to healthier water? Had they died from the lack of oxygen? There was no way to know instantly. The river kept its secrets. Researchers set out crab pots, wire cages baited with fish, to see what they could catch. This was the moment that would decide everything. When they pulled up the first pot 3 days later, hearts were pounding. As the cage broke the surface, water streaming off the wire mesh, they saw movement. Blue claws snapping, shells scurrying. They checked the tags. They were the hatchery crabs. And they weren't just alive, they were feisty. The mood on the boat shifted from pessimistic to cautiously optimistic. They had survived the drop. But surviving the first week is one thing. Living long enough to make a difference is another. 2 weeks in, the underwater cameras started picking up something fascinating. The crabs were behaving territorially. They were defending small patches of the river bottom, fighting off other crabs and establishing homes. They weren't acting like lost tourists. They were acting like they owned the place. 3 months later, the real shock came. Researchers were conducting routine surveys, pulling up nets to count the population. They found something they didn't expect: baby crabs, tiny microscopic versions of the adults. But these weren't the ones they had released. These were brand new. The released females had mated. They had produced eggs, and those eggs had actually hatched and survived in the polluted water. The next generation had arrived. This was a game changer. It meant the released crabs weren't just a temporary band-aid. They were a seed population. They were restarting the natural cycle of life in a place that was supposed to be dead. The scientists were stunned. They expected survival, maybe, but reproduction? That was the holy grail. And get this, the water itself was starting to change. Sensors placed in the river began to pick up subtle shifts. Dissolved oxygen levels near the bottom were creeping up. It wasn't a huge spike, but it was there. The amount of organic sludge in the sediment was going down. The crabs were eating it. They were consuming the fuel that would normally rot and eliminate the river. They were literally eating the pollution. It turns out 10,000 hungry crabs can do a lot of cleaning. They were devouring dead fish, decaying plants, and clumps of algae. They were acting like a biological vacuum cleaner. And because they were moving around so much, digging into the mud to hide, they were churning the sediment. This might sound messy, but it is vital. It releases trapped gases and allows fresh oxygen to penetrate the soil. They were tilling the garden of the riverbed. 6 months after the release, the underwater grass beds, those vital nurseries for all aquatic life, started to look different. In the Lafayette River, grass coverage had dropped to about 15% of what it used to be. It was pathetic. But after the crabs moved in, the grass started expanding. It jumped to 23%. A year later, it was over 30%. The crabs were clearing the way for the grass to return. By eating the algae that coated the grass blades and blocking the sun, they were giving the plants a chance to breathe and grow. And more grass meant more oxygen, which meant healthier crabs, which meant more cleaning. It was a positive feedback loop. The ecosystem wasn't just stabilizing, it was waking up from a coma. But the most incredible part was what happened next. The return of the crabs didn't just affect the grass and the mud. It sent a signal to the rest of the ocean. Something big was coming to hunt. The domino effect you never saw coming. This is where things get really interesting. Scientists call it a trophic cascade. It is basically a domino effect that starts at one level of the food chain and ripples through the entire system. And in the Lafayette River, the dominoes were falling fast. The crabs weren't just eating trash, they were changing the behavior of everything else living there. Before the release, the river bottom was dominated by small, unchecked fish and worms that had no predators. They were stripping the resources bare. But when the blue crabs arrived, the bullies of the playground were back. The small fish started hiding more. The worms and clams that were being overeaten suddenly had some breathing room. Why does that matter? Because clams are filters. When the clam population rebounded because the crabs were keeping their predators in check, the water filtration doubled. More clams meant clearer water. Clearer water meant more sunlight reaching the bottom. More sunlight meant more grass. It is all connected. The crabs were the generals directing this entire operation without even knowing it. And then, the big guns showed up. Researchers started spotting large predators in the river. Rays, large drumfish, and even small sharks began venturing into the Lafayette. Why? Because the buffet was open. The crabs had become a food source for the larger animals. You might think, oh no, the crabs are getting eaten, that is bad. But no, that is actually perfect. The presence of top-tier predators is the ultimate sign of a healthy ecosystem. It means the food web is rebuilding itself. It means the energy is moving from the bottom to the top effectively. The river was supporting complex life again. It wasn't just a stagnant pool of algae anymore. It was a hunting ground. By 2015, Virginia expanded the program. They saw the undeniable success and released 50,000 more crabs across 5 different sites. The results were consistent every single time. Within 6 months, water quality improved. Grass beds expanded. Fish populations rebounded. Maryland jumped on board in 2016. Even the federal government, which usually moves at the speed of a snail, started throwing money at the project. By 2020, over 500,000 juvenile crabs had been released across the bay. The data was irrefutable. Every single metric, nitrogen levels, phosphorus concentrations, fish diversity, improved in the areas where crabs were added compared to the areas left alone. The science was settled. Active restoration worked faster than passive protection. But let's not get carried away. The Chesapeake Bay isn't magically fixed. There are still dead zones. There is still pollution dumping in from farms every time it rains. 10,000 crabs, or even 500,000, can't stop industrial runoff. But what they proved is that the ecosystem has a fighting spirit. It wants to heal. It just needs a kickstart. The success of the crab release forced scientists to rethink how they handle environmental disasters globally. For years, the standard procedure was clean it up, then wait. But waiting takes too long. Sometimes nature gets stuck. It reaches a breaking point where it can't recover on its own because the key players are missing. The crabs showed us that sometimes you have to intervene aggressively. You have to throw the key players back into the game, even if the field is still messy. This concept is now being used all over the world. In California, they are looking at sea otters to save kelp forests. In the Gulf of Mexico, they are reintroducing fish to coral reefs. The Chesapeake experiment became the blueprint. It proved that biological restoration is just as powerful, if not more so, than chemical or mechanical restoration. We realized that we don't always need to build a machine to fix the environment. Nature has already built the perfect machines. They have 10 legs, snap at your fingers, and love to eat dead fish. We just needed to get out of their way and let them work. But there is still one big question left unanswered. Why this changes everything we know. For decades, we operated under the heavy assumption that we were the only ones capable of fixing the mess we made. We approached ecological collapse with the mindset of engineers and economists, convinced that the solution lay in better technology, massive influxes of funding, and bigger, more efficient industrial filters. We thought we could build our way out of a biological crisis. But the blue crabs taught us a humbling, necessary lesson. We aren't the saviors of the planet. We are simply the ones who broke it. The real repair crew was there all along, submerged in the muck, waiting for a chance to clock in. We didn't need to invent a new machine. We just needed to get out of the way of the one nature had already perfected. The recovery of the Lafayette River serves as a profound proof of concept. Ecosystems are not as fragile as our anxieties suggest, but they are infinitely more complex than our current science can fully grasp. For too long, we treated the bay like a giant chemistry set. We obsessed over metrics, trying to manually balance the pH, tweak the nitrogen, and inject oxygen levels as if we were maintaining a swimming pool. We failed to realize that we should have been treating it like a living organism, a patient that needed its immune system reactivated, not just its fluids flushed. The crabs were that immune system, acting as the liver and kidneys of the river, filtering out impurities through the simple act of living. Today, the Chesapeake Bay stands as a testament to this shift in thinking. It is measurably better than it was in 2012. The blue crab population has stabilized, clawing its way back from the brink and bringing the rest of the food web with it. The water is clearer, allowing sunlight to reach the riverbed and encouraging seagrass to take root once more. To be clear, it is nowhere near the pristine, teeming estuary it was 300 years ago. And in our modern world, it might never reach that state again. It is scarred and it is changed, but it is alive. And in the Anthropocene, survival is the only metric that truly counts. However, this success raises a controversial, almost uncomfortable question for the future. If we can jumpstart a dead river with crabs, what are the limits of this approach? We are standing on the precipice of a new era of ecological intervention. Could we reintroduce specific species to the Amazon to accelerate reforestation? Could we deploy colonies of mollusks or genetically adapted bacteria to clean up massive oil spills, replacing chemical dispersants with biological agents? Or by attempting to manage these intricate webs, are we playing God with systems we still don't fully understand? We are just scratching the surface of what biological restoration can actually achieve. The blue crab experiment was a gamble, a desperate shot in the dark that happened to pay off. But it opens the door to a wild, unpredictable future where we might finally stop trying to conquer nature and instead learn to use nature to save nature. Are we missing the obvious solution for climate change and pollution? Is the answer swimming right under our noses? Tell us in the comments below. And if you want to see more incredible stories about how nature is fighting back in ways science can't explain, make sure you hit that like button and subscribe. You do not want to miss what we uncover next.